Okay, welcome everyone to the next NASK Science seminar. It's my pleasure to introduce to you Dr. Alessandro Crimi, who is a biomedical engineer and health economist who alternate his career between neuroimaging and healthcare management in low-income countries. He did his master's at the University of Palermo and then obtained a PhD in machine learning applied to the neuroimaging at the University of Copenhagen. And then he did his MBA in healthcare management at the University of Basel. His postdoctoral research tract is very broad. He worked at the French Institute for Research in Computer Science, the Technical School of Switzerland, ETH Zurich, Italian Institute for Technology, and University Hospital of Zurich. Among these, he also worked for 8 years in Africa, in the African Institute of Mathematical Sciences in Ghana, and you were teaching there machine learning in medicine, as far as I understand. And he conducted also projects there related to prenatal care, diabetes, malaria, HIV, and so on. And currently, he's the head of the neuroimaging lab at Sano, working on finding novel biomarkers related to brain diseases. So, welcome. We are happy to hear your talk, and the floor is yours.
So, thank you for the invitation, and actually I'm impressed you know every step. And I'm not 80 years old, so while I was working in Switzerland, I was going to Ghana. So these 9 years in Switzerland, 9 years in Ghana were in parallel. I'm not 80 with doing all this stuff. So yeah, as I was saying, thank you for the kind invitation and to be here at the NASK. And I currently, among all the stories that have been mentioned, so I'm here because I'm the group leader at Sano for the medical imaging group, although we are mostly focusing neuroimaging. So from medical imaging, we were supposed to do many other things, but we work mostly in neuroimaging. And I'm also assistant professor at the AGH, where we teach the course of the neuroimaging. And yeah, it's working. So a bit of, for those who doesn't know what is Sano, because Sano is not, technically is not part of AGH, is an independent institution, is a foundation, is an independent research institute, although we are still located inside AGH. And we are in these colorful yellow windows that they look colorful from the outside, they're more annoying in the inside. And this is currently my team. So we have like four PhD students and 3 postdocs, and we are even allowed to have a dog from AGH. Usually it's not allowed, but we managed to have it for psychological support. Is the dog of one of the PhD students. Yeah, and I'm going to present some of their work and what we do from neuroimaging. One trend that we are mostly interested is related to network, which I assume is something, networks are something that is involving other fields, including also people here in NASK. So if you work at cybersecurity, I imagine you know a lot more than me about how internet is used as a network and the security involved and packet transmission than many other people worked in computational for computational representation of gene interaction, proteins, and all these things. So this is also a network. And probably after COVID, we all are epidemiologists, and we all know that people are networks, and disease is spread. So this to try to make a bridge between what I'm going to tell you and other fields that probably people here at NASK are doing. And so what we are interested mostly is this representation, which I'm going to briefly re-summarize how we get there. So we consider the brain as a graph. And apart from this fancy and cool representation is that we try to relate this representation as an intermediate phenotype, that then we correlate either with some disabilities or some behavior or something, or maybe we go on the opposite direction. We try to relate it to some genetics or genomics. So I will also mention some study we try to like kind of close this loop. And yeah, and like everybody, we work with neural networks right now, but so there are people working a lot of neural networks which were inspired by the real biological networks, but we're also using the artificial one. So we are like that. We're like using them like to give back a favor, to use them to study the physical network using the artificial networks. It might be a bit confusing, but I want to say that we are mostly interested on what we can find as a biomarker. So if one day we find something that is not cool with neural networks, we are okay dropping this. We are not very obsessed supporting AI as a buzzword. And yeah, one example typical case is that we end up in this representation of brain networks, for example, for some group of patients with Alzheimer, and then a group of people healthy matched control, like people of the same age, and we try to distinguish them. So either for help some classification, some aid the work of some physician, or maybe to find some specific difference that can be some biomarker that can highlight give some insights to physicians. And when we say this, and now I start introducing how we get there for those who are not familiar. I know some of you are very familiar, but I imagine some people are hearing these stories for the first time. So we are interested mostly in three major connectivity. So structural, functional, and effective, which I will review them very quickly. Then if we have time, I will spend some time in the effective. So structural means like literally the physical structure, the axon bundles in the brain, and functional is more like the relationship between some region of the brain in time. And effective is more fancy and more complicated things, and we actually still have to realize it really is useful, which is more related to the cause and effect in time. So if there is some activity at some point afterwards in another region. So with the structural, we're starting super quickly is because we at least it can be reconstructed in many ways. If we look at it as using magnetic resonance imaging, there are some modalities, like one is called diffusion, where we can track water in some tissues. And particular things is that normally water is the water particles, hydrogen, are moving around, while instead if there are some membrane like around the axon in the neurons, they are like bounded by the membrane, so they kind of start having some shape. And also just a reminder, like the cells, so the main cell body is in the gray matter, that's why it's darker, and then the axons are in this white part. So what we're substantially going to be able to do it is that we kind of are able to distinguish where there are these rounded parts that are probably in the gray matter from the elongated part in the white matter. And the simplest thing that usually people were using it was with diffusion tensor, where they were like kind of reconstruct the probability for each individual voxel in the MRI, and they were kind of able to detect this shape if it was elongated or it was more isotropic, just to understand if you were here at the green matter or inside more in the white matter. Just this was super high level. It's not even used because most people actually found a lot of problems with these fiber tensors. So there are more sophisticated and more advanced and more useful approaches, because there is a problem with the diffusion tensor when there is fiber overlapping, that they tend to be also isotropic, while in reality it's just a crossing. So sorry for the meme, just to tell you that this is just was the simple things, but maybe there are more useful stuff that are more advanced that I'm not going through. Then something I used actually we were trying when I was in Zurich was more the physical tracks. So we were like injecting some adenovirus and we were tracking, and it's super annoying because you need stereotactic things, all this stuff, but plus also you need to kill the animal. So it's not very, it's good for basic research, but not for diagnostics in humans. But anyway, just with the, let's say with the spaghetti neurons, what we were getting was, in some cases, few years ago, we were even able to find some kind of biomarker just with some sparse discriminant analysis. We were finding what were, according to a sparsity of the features we were passing, like was useful for the classifier. We were kind of able to detect relevant features. And in this case was some mice that are typical for autism. So they resemble how the behavior of the mice in humans are for autism. And it's a wide-known model because it's known that they have more corpus callosum and less fibers, ipsilateral fibers. And we kind of were able to find this automatically. But it's still a spaghetti, so it's still not so practical. So what we do to move from spaghetti to something a bit more friendly, like this, is that we combine the spaghetti neurons to some kind of anatomical parcellation, like this is an entire word, like how you parcellate the brain. But let's assume we have a good parcellation from somewhere, from Brodmann area or Brodmann or other more recent things, site architectures or so on. So the idea is that we know that one region, so this is an atlas, and we consider all the fibers in this region. This is simplified as a node. Okay, I assume you remember it. So there is this is simplified as a node, and this is this entire region is simplified also as another node, and all the fibers going, let's say, from this to this, they are I wanted to go back, is not? So one region is now one node, and all the fibers going from, let's say, this red one to this green one is just one edge. And how many fibers you have there is the size of the edge, and how many fibers are entering one node, so it will give you like the degree of the node. So far is this, I assume you all know this. I hope online if it's not, are there a way for them to ask questions on the?
Yeah. It's possible. We follow the discussion on YouTube. If there is any, we'll let you know.
Okay. So if someone wants to ask questions, is the right moment. Because I'm going to the other one. So okay, this worked. So the other one is the functional connectivity. So instead of measuring the physical fibers, like in time, there is another way of measuring the hemodynamics within one specific voxel. So let's imagine I'm looking at this voxel, red one here, and in time, I see those changes, and the blue voxel in time, I see those changing in the signal that represent changes in hemodynamics. And people have noticed a long time ago that sometimes, maybe I ask you, do you see anything? Because this is representing 3 brain regions. So this ipsilateral, the posterior cingulate cortex and medial prefrontal cortex here are like this yellow, orange, and blue. Do you see anything interesting?
So once the orange and yellow are elevated, the blue is decreasing, right? It's always the opposite.
It's the opposite, and also these two tend to move together.
Yeah.
So what is the observation is that this is like anti-correlating, IPS is anti-correlating, and the other two are correlating. So, and this kind of, okay, this was the default mode network. That's something widely known. That is like the basic network that is always active in a person. So it's continuously doing this kind of stuff. But like imagine you generalize to the entire brain the story of correlation. You now have a big correlation matrix. And from this correlation matrix, you can also make a graph. So this is another way of constructing those graphs. And then we use those graphs again to either do some automatic classification or we look for a biomarker and so on. This one, I just mentioned it. I don't know if there will be time, I will go back because this might take 1 hour just to discuss about this. So there is the further one that is the effective connectivity. So effective in the sense that the activity in one region is causing some effect in another region afterwards. So when this like open up the entire story of the brain as a dynamical system and so on, and plus also why is this appearing? So it's appearing because they just correlate, or probably there is an underlying tracks. So it's also related to the structural, or maybe it's not that simple. Maybe it's related to structural, but there is more like a ping pong. So before activating here, maybe it's passing first here, then it's going here. So it starts making stories of the studying the brain a bit more complicated. I will come back to this if we have time at the end, but just these things exist. Okay, so that's what I was, these things, the first things that I was trying, okay, this was published in 2021, but actually it's a very old study, like I think 2013. We were using SVM, there was no deep learning, so imagine which time I'm talking about. For a series of reasons got published later. But what we did was that substantially for the same group of subjects, we reconstructed the structural connectivity, functional connectivity, and one representation of effective connectivity. And we had this for young teenager with autism and matched young teenager that have been assessed without autism. And we were trying to see if there was some way to discriminate. And there was, with SVM, there was a statistically significant discrimination. Plus, we were looking what SVM was finding. So we found some let's say, connections that were relevant. And these were from the structural, these were for the functional, and these were for the effective. I don't go too much in detail. So these were already known in literature. So we kind of confirmed what other people knew. And what we noticed that the effective was kind of more or less making a mix of these two. But anyway, this was just, we were just using the metrics in a most stupid way. So we computed the connectome, and we were using the entire connectome as a feature. So there was nothing, of course, in those days also, even with SVM, there was no deep learning, nothing. So now we are more aware of what we do. So instead, since I moved to Sano, we did something a bit different. So this was from with Samar, which was actually, I was at Sano already in Krakow, but she was still finishing her PhD in Cape Town. So this was it's a cross moment between South Africa and Poland. So what we wanted to see was that we collected we reconstructed the structural connectome of people with Alzheimer and healthy elderly people. And the idea was that within 2 years, the people with Alzheimer, they had bigger changes because, of course, the neurodegeneration is stronger. So they say if this is the connectome of someone with Alzheimer at 60, then at 62, they already have some loss of connection because of the neurodegeneration. While maybe the same person, same elderly people, when they are like 60, from 62, not much is changing. So of course, we get old and something is probably deteriorating, but nothing that is visible. While instead, if you have Alzheimer, you kind of, we should be able to detect these kind of changes in the connectome. Of course, this was also obvious, so was nothing super fancy, so we kind of proved that there is this difference. But also what Samar investigated was just also the correlating those kind of changes with some genome, because we also had the genetic data from these people, so we could see if those changes in the networks can be used as a feature for a genome-wide association study.
Just one question,
yeah?
if I may. Would you say that in dementia the changes in the connectome would be similar? Or at least it will be like intervening state between normal and Alzheimer? Or nobody actually looked at it?
I always thought that, so dementia is the overall deteriorate group of disease. Alzheimer is one specific disease.
Okay.
Dementia is more broad definition.
Yeah, that's what...
Okay, sorry, that was my mistake then.
No, okay, so maybe...
Because I thought dementia is just a starting Alzheimer.
It's
but so Alzheimer is
one symptom.
Oh, okay. So sorry.
But you can also have dementia just because you're getting old.
True.
Yeah, that's a good point. We never look at that, but I assume it's happening the same. Probably the thing is that if you have Alzheimer, probably this is happening when you're 65. Dementia, because you're getting old, is probably when you are 84, 86, 88. That's happening. We all have this. So there are this misfolded protein. We all have it. It's just that they get more and earlier. And yeah, I will get to this protein. I have another thing. So what she did was she analyzed these changes in the networks, and also she tried some graph metrics like efficiency, sensitivity. So there are ways to, they are coming not from neuroscience, they're coming from complex networks to summarize the network in with the scalar value. And she looked at this genome-wide association study, and she found 4 were significant. One is the most known, one of the most known risk genes, so nothing new. So this is a gene, if you have it, increase your chances like 70% to have Alzheimer. And the other were kind of relatively new, and we still don't know what they mean, but they were related to cholesterol and iron transport in the brain. So this was more basic things to maybe give some insight, someone who can look at it, well, iron, and I'm not a neurobiologist, so I assume they might know more what does it mean, what this means. Yeah, another thing that we did more recently was actually looking at the network not as a feature, but more like as a backbone. Because there is this explanation that Alzheimer, Parkinson, or even normal frontotemporal dementia because you get old, or other disease, they appear because you have some proteins that they move around in the brain, and they accumulate more and more and more. And in the case of Alzheimer, there were 2. So there are 2. So one is amyloid beta and tau filament. Amyloid beta is generally at the beginning, and tau filament is what you get afterwards. So this is, first you have this one, this purple one, and then you have this one here. And generally when this is popping up, you have a lot of serious disability, while this one is probably some kind of mild cognitive impairment, and people not even pay attention. So that's the thing. We should kind of detect people here and try to do something. That's why most of the study and therapies don't work, because they try to do something when it's too late. So what we wanted to see if we can predict the spreading of these proteins. So we had the structural connectivity as a backbone this time, not as a feature. And now our features were the misfolded protein deposition. So we know that let's say someone at, as I was telling you, you scan at 60 and then at 62, and you can see where they have this deposition at 60 and when they have it at 62. And this is not just to correlate simple colorful pictures, it's just also because we know that when you start having a lot of the tau filament, you probably have a lot of disability. So it could be a way to predict the future disability of people with dementia. And of course, we were not the first doing this kind of analysis. So these are the 2 state-of-art model. One is from Iturria Medina, which is called epidemic spreading model, and the other one is from Raji, the nectar dynamics model. And we tried two things, comparing them if we were better or no. One was just considering some autoregressive model, which is some kind of old school statistics or old school machine learning, and then graph convolutional networks. So these two that they've been considered the state of art for this kind of prediction. One is inspired to epidemic spreading, so like these things I was telling you of COVID spreading through people. So, but instead of measuring how a disease, infectious disease is spreading between people, they were using brain regions. So this is instead of one person here. So this will be one person passing, let's say, COVID to this other guy, then passing COVID to this. They were measuring this in brain region, and the other one was more like coming from physics, like they were essentially making like finite element simulation with spreading heat equation. So they were considering the misfolded protein as a heat spreading on the network. So we recreate the code of these two, and we also implemented our two possible solutions. We wanted to see who were of all these four better predicting, given some initial deposition, who were better at predicting the misfolded protein deposition after some years. And yeah, the first one that we tried is kind of simple. I don't know if it's obvious to. So we were considering Y as the current protein deposition, and then we were estimating in a loop like a cross-validation loop if there were some regressor that can predict the protein deposition afterwards. So this is just really just this equation, make it in a loop to optimize this coefficient. And maybe I will spend more time what we mean with graph convolutional network. So the idea of normal traditional neural networks, you have a bunch of layers and a bunch of convolutional filters, and generally it's for images. So we consider like one filter as one function, and the pictures we are kind of studying with neural networks are the other function, and we try to optimize this the convolution of the two. But it's kind of it's working very well. Like we know, NVIDIA is making millions. So with graph looks appealing, and we might think we can do the same thing, but there are a series of issues. So the first issue is that a picture, each point has the same distance, so they're kind of in Euclidean space, while a graph, like here you see the grid, they are equidistant, so you can kind of consider it a neighborhood in a Euclidean manner. While in a graph, you have something like that, it's probably non-Euclidean, so you don't know this guy connected at this distance, how you can measure. Plus, it's not so regular, so here a neighborhood is always 9. A neighborhood is always 9 points, so 1 dot and 8 around, while here you can have 4, 3, 5, 100. So it's making things a bit more complicated. Also, what does it mean, convolution in a graph? So I will actually, at the beginning, I'm not even sure if we should call it any more convolution. So it's what we consider convolution is more something like an aggregation of the neighborhood. So because then this is like what we will do as a corresponding of the convolution in the picture. So we kind of consider the value aggregate in the neighborhood, and then also how this aggregation could spread, because of course here you have one aggregation, then now this node you have another aggregation. So it's more message spreading on the network. And of course, you have some weight parameters that you will optimize during the training. This is just, as I was saying, for the diffusion, this is just the beginning, because there are plenty of other variation. And actually, in those days, we try only these three. We didn't try the attention network. We tried in another case, and actually, none of these were working because these things have a lot of problem. lot of problems. I will show you another case where also didn't work, but at least we understood why it was not working. And then one case where it finally worked. So this is a typical result of a prediction. So this is one person. We are showing for each individual region the error of reconstruction of prediction, and the GNN actually was the worst, and the other three were kind of very similar, and actually, but paradoxically, the one who was performing better was the simplest, the autoregressor. And was performing better, we know why, because the spreading, they tend to always increase, while biologically, as I was telling you, there is this thing that at some point in the brain, this misfolded protein, they stop increasing. So this somehow the autoregressor coefficient was able to predict this case but these other two, they were always trying to increase. So in those cases, the autoregressor actually was working better. I don't know how much you can understand from this picture, but if you look at it, that we noticed that comparing all four, the worst was the graph neural network, and then the other three, they were kind of similar, but in some cases, as you can see, the autoregressive model was performing better. Yeah, this is what are our conclusions. So that you can have an alternative way of predicting misfolded protein, either with traditional multivariate autoregressor model or with graph networks, but the graph networks was the one actually not working very well. I have another case, and we also finally understood why this one was not working very well. So this is the other case, which is another project, carried out by another PhD student, Joan Falco-Roget, with some data from Ghent in Belgium. And it's about brain tumor, and I assume you know this part you at least you know. So someone is before the surgery is undergoing some scans and of different types, functional, structural, and then the surgeon is making some planning, and then they have to cut what they have to cut, maybe what they think is from gadolinium activated or so on, and what was our insight was that we wanted to make a prediction of what's going to happen after surgery and after recovery. Why? Because anyway, the surgeon will make his own decision, like what to cut or what to do, but at least we could show what is going to happen afterwards to give some insight of how we might look like after he's performing the surgery. We want to show him, this is most likely the future. Think about, because maybe from what you are resecting, you might cut something and this person has aphasia, is not able to speak, or has some motor deficit, and so on. So this was our insight. If we can, let's say, we have some kind of adjacency metrics of the structural connectivity before, we predict how it will look like after, and we show to the surgeon, and maybe it's giving him some insight rather than just looking at the current things. We tell him the future, maybe it's giving him some insights. And yeah, this is what we try to do. And now what the idea is that from one graph, we should be able to predict the future graph. The first thing that came to our mind was graph convolutional networks, because also there is a group that is only doing this, graph convolutional networks in brain, and they have a lot of papers. We try all some of they have it online, but none of this worked. And why it didn't work? Because they were testing it on healthy subjects or in some controlled cases, while our data set was super heterogeneous. So some people were having a brain tumor here, some people were having a brain tumor here, so it was not that trivial to make this training of these graph convolutional networks. Plus another thing that also gave insight why it didn't work in the previous studies, that the story of the message passing when you have a very large network doesn't perform very well. So there is also we were using a small data set, so we had like 20 subjects with brain tumor. So this combination of things makes graph convolutional networks not working very well. So if you have a connectome with 300 nodes and 20 subjects and you use graph convolutional networks, for other machine learning things, maybe you have an easy solution for this stuff, but for graph convolutional networks, because you have the story of the message passing is degenerating very easily. And in fact, in the end, what Joan ended up doing was he had some kind of Bayesian, like we did, the autoregressor was working better. With this, in this case, he had some Bayesian linear predictor that it works better than the graph convolutional networks. So I'm sorry to disappoint everybody. We're like excited about graph convolutional networks, but sometimes, depending which problem you have, is better something very, very simple. I don't know if you can see. This is so this was the subject connectivity, and this was the I think the reconstructed. So and I'm pointing some area near the tumor. Some prediction were accurate, and here you see the. Do you see the residual error? So it was kind of working well, just this simpler linear predictor. Now, one case that where finally this GNN gave us some hope. So was another study by Szymon Mazurek. Was that we were we had a data set for people with epilepsy, and our idea was that we could make like a functional connectivity of people with epilepsy and predict when before they were having a seizure. So not seizure detection, because that's too late. So it's not practical, but at least if we can classify the functional connectivity of the 10 minutes before in the pre-ictal phase, because there start being something. So maybe ideally you have, I don't know, something you're wearing. So while you're driving, these things, in 10 minutes, most likely you have a seizure, so maybe you park. And then if the risk is over, If then in 10 minutes you have a seizure, at least you have 10 minutes to think what to do. So, and we had a very long recording, so 960 hours. The sample size was small, but we had a lot for each people. And they were mostly young people or children. And so these were not an MRI study. This was from EEG. So EEG is kind of, I will not say the same thing of fMRI, but for functional connectivity, you can see it as the same thing. So you have a series of electrodes, so that are kind of fixed. So this will be the equivalent of the atlas. And for each of these electrodes, you have a time series, so we can do functional connectivity, or at least studying this signal in time. One thing that Szymon introduced that actually it helped us a lot, I have it here with just 5 nodes, was that instead of just, I don't know if you remember the story with how we consider the convolution in a graph, we used something additional, which is the attention. So these were graph attentional networks. Attentional in the sense that for each edge, there were an attention coefficient that was estimated during the training, and this kind of help us to get better results. So with graph attentional network, apart that we were able to spot the connections that were more significant, also improve the results. And yeah, these were in individual fold, but I mean, even doing further cross-validation, the results were even if they were getting a bit worse, they were still super high. And these were the mapping of the most relevant connections between the brain, helpful for detecting this kind of stuff. So it was from F3 to O2. So the connection between these two areas. But at least it gave us some hope that this graph convolutional networks, we can use it for something and it's not just exploding. This is a different data set. We changed, okay, it's a better model plus there was more data to train. So we are not sure what helped in both cases. But anyway, finally we get something that makes sense. And another thing, so I guess now you have seen all this dystopian situation. Also, this guy, I assume you've seen in social media, Vision Pro is exploding, and now we are even having more like Simpson predicted years ago, where the world is. I haven't seen anyone in Krakow. Did you see anyone in Warsaw with this? Not yet.
So far, you cannot buy them.
Huh?
So far, you cannot buy them in Poland.
Officially, they are not.
Until when?
Not really known. I mean, the only people in Poland who got them had to travel to USA.
So we are safe for a while. Anyway, this is not. I don't want to criticize them because actually we were. We did. What we did with this, if you remember the story of the surgeon, so we, with someone at Sano in his living room, plotted in AR the connectome. So imagine if these things I was telling you of the prediction of how the connectome will look like if we see it in augmented reality. We discussed with some surgeons in Palermo, and they were like, yeah, let's try tomorrow with the patient. But the ethical approval, yeah, sorry. So, so we slowed down. So we had to do the ethical approval. We are starting now. So but we kind of developed. So no, this is just the living room of. Okay. So the idea was not with the vision. At that time, the vision was not even in the market, but we thought it is more friendly that a surgeon has a tablet. So is doing something like this. So he's marking, and then with the tablet is overlaying either the current tractography or current graph or the future one, the one I was telling you that Joan was able to predict. And oh no. I should have it here. So this is a real plastic head. So the idea is that we have to match this. We have to dock. We use like this shape you see here. This is I think it's the mobile phone of Joan actually. So this is the mobile phone of Joan. So he is able to dock, and now once he docked, he could load all this structure. I mean, still playing. I know again. You have to watch all of it. So I think this is the mesh from the T1, and then this is the connectome. I think I don't know if we already are that load current or future connectome, but probably we should add the current or future connectome, and then also the trucks, like here. Okay, we have the prototype. We now are supposed to do in Italy the real test and so on. Okay, and then the last thing that this thing is. I don't know how to stop it. So one more thing that we did that is also like let's say cool, but we have to. I will tell you the truth, what is cool and what is useful. So we, given still this representation of the brain connectivity, there is one common issues, like if we can cluster these things, if we can make like graph partitioning. And there are these things have been studied for, I don't know, 50 years or maybe even more. And what we wanted to see, because in the end some of this stuff is based on optimization, and the insight was that if we are running on D-Wave, which is like the quantum optimizer, maybe is producing a better optimization. And so we ran this first for one toy case study, then the one partition of the brain and one more advanced partition of the brain. And what we noticed was that for two cases were practically the same. So if you run the one Louvain modularity in a normal computer in quantum, is probably the same. You just spend a lot of money, a lot of time in the quantum. While if the parcellation of the brain is very complicated, the quantum will start showing better partitioning given some ground truth. So what we saw is that for some cases, this story of the quantum computing, it's useful. It's another thing overly hyped, so not everything is really useful, but in some cases, if the complexity of the network is very, very big, these kind of optimizations are useful. And yeah, I think I showcases enough case study. So the story, the overall idea is that we have different ways to reconstruct the brain as a graph. And once it's a graph, we can do many things, from stuff we know from complex networks, use them as features for machine learning, applying graph convolutional, doing simulation on them, and many other things. So we are still investigating what else can be done, and I'm ready to take questions. Just some small advertisement. So Sano, as I was telling you, is located inside the Agea campus. It's an independent research institute, and we're always open-minded to find collaboration partners and so on for the rest. And yeah, as you mentioned at the beginning, I had another life going back and fro. So this is I had some fun with goats and other things, and pregnant women, and so another things I have in Italy is an association to promote entrepreneurship for people African arriving in Italy, let's say. I think there could be the same usefulness here with all the Ukrainian, that instead of looking for a job, maybe they can also be focused on entrepreneurship, but we will see. And at the moment, I'm trying just in Italy. And yeah, and that's all. I don't know if I have time. There's questions, or I can show you another. I can bomb you with another thing.
So maybe we will take questions for
now,
okay. And then we will continue with the next topics. Well, thank you very much. We don't have yet questions from YouTube. I guess people will, but we had at the peak over 50 people listening on YouTube, and there's still plenty of them. So I encourage everyone on YouTube to ask questions in the chat. I will read them. Are there any questions from the audience over here? I have a couple, but I can give you the start.
Hi, my question is about augmented reality and the docking problem, because you have shown how to map the visualization of the brain on this plastic head, or something like this. But the docking has to be done to the fraction of millimeters, very precisely, if it is going to be useful for a neurosurgeon. How are you resolving this precise docking between the augmented reality and the actual reality the neurosurgeons are going to work with?
So these are some tricks that Bartek. So first of all, we have the idea that this is done when the patient is not moving. So they position it, and then it's not. So it's already anesthetized, so it's not should not move. And then the docking is not done automatically. I don't know if you, the video is probably.
Yes, yeah, I've seen that.
We move it, we scale it, we move it, and then it's locked.
Yes.
And then all the other structures appears according to the reference of the head.
Okay, so it is for now it is done manually.
Yeah, and we are not even sure we will ever manage to do it automatically.
Because if it is done manually, I am wondering.
If they move the table.
I'm wondering about the precision of the docking. Because if your docking is, for example, a few millimeters off, and the neurosurgeon is going to use it, and he will operate a few millimeters off the target, well, it's not a good idea. The precision of docking is essential if it is going to be used for surgery.
Well, combining this with something that BrainLab has, like the stereotactic clamp and the RFID system of RFID.
Well, yes, something like this, but because, for example, if you are docking, let's say we are fusing the MRI with CT, we are using certain anatomical landmarks like, I don't know, ACPC and so on. In this case, we do not have something like this, so we have to dock, let's say, by hand. And what I'm thinking is how to obtain the maximal precision in this docking, because it is essential.
Mm-hmm.
For usability of this in practical
we started
approach.
Now this thing.
Yeah.
So this is the first thing was done docking by hand. We can continue to collaborate, and maybe you have ideas how to do it automatically. But we were even impressed that we could do it.
Because you were talking about this Vision Pro and so on. We are thinking in this area also about using some kind of augmented reality in the neurosurgical operation theater. But well, it has to be somehow we must obtain the maximal docking precision.
I agree, and this is how far we got. We don't. Actually, we were impressed. We managed to put together all the pieces.
Exactly.
So I have a question regarding the EEG. I'm not very familiar with EEG, so my question might be really silly. But as far as I understand, so the problem with MRI is that the experiment is quite expensive, even though if you run this prior to the surgery, and you need to wait a lot for the radiologist to annotate, to prepare all the documents for the doctor. And actually, it seems that by detecting different changes in the connectome networks graphs for a given person, you can detect very early on some degenerative diseases like Alzheimer's, for instance, right? That you can see that something is altering. So it's all cool to do when you have an MRI, but can you go with EEG? Like, does EEG have a resolution that is sufficient for such studies? Because that would be, I guess, less expensive and more accessible for in many countries.
Yes, definitely yes, if we do it. But this is the 1020 systems. There are still systems with more nodes, but you don't get the same resolution of.
Yeah.
I guess that. Well, for wait, no. For Alzheimer, there is actually. I think there is a startup in Slovenia which is trying to prove this.
Okay.
So it's not us, but I met them on a conference. They were showing that they were trying to make from EEG connectivity analysis for Alzheimer and predicting.
Cool. Yeah,
so there is a way for these. The motivation is that seizures in people in the real world, and it's like a heart attack. If you have an heart attack while you're driving, then you kill not only you but other people. So this was more, say, urgent.
Yeah, because I was thinking also how tightly you could pack those electrodes on the surface of the head to record even smaller patches of brain activity.
But now there are a lot of trends of patches in the headset. So
okay.
we are improving. This is the clinical stuff, and of course it covers more stuff, but definitely you cannot wear these things. So there are many people studying like a headset that they can be wear the entire day.
That's cool.
But of course you are only, let's say, then you are limited. Then your signal is here and here. You have no idea.
Yeah. And then there's Elon Musk, who has Neuralink.
Neuralink, yeah, that's another level.
We will steer our cars with thoughts. Okay. Are there any more questions from the audience? I don't see anything from YouTube. So if there are no further questions, thank you very much. It was a pleasure to hear. I'm really...
Another funny thing I can tell you that we are trying to fight is that it's more a curiosity. About EEG, and since you were mentioning this thing in Africa, I tell you. So the EEG devices are kind of racist. I tell you why, because our hair is not the same as their hair. So they have really this curly and scratchy skin. So if you put like a European or Western world EEG, it doesn't read very well. So there was one lady say, now I'm designing the African electrodes because these things don't work. It's just a fun fact, okay?
Curious, yeah, curious. I wouldn't thought that the type of hair would matter, but well. Okay, so as far as you told us, you have one another story. So let's say the official part is over, but for those of you who want to hear more from Alex, stay with us and we will continue for a couple of more minutes, I guess.
Do you really want it, or?
Sure.
Because I'm okay. Because we are going esoteric. Okay, actually this is my preferred topic. So we are back in the... So the story of the... as I was telling you, of causality, because people are both interested either that we make another kind of biomarker, so there is not just structural or functional, or we also have this effective, because there is a way with another